when you look at two directions defined the gradients for PC1 and PC2, you said there is no meaning whatsoever associated with these directions?
Yeah, so I'm not saying there's no meaning associated with direction, but one has to be very careful about the meaning. So for instance, in that case where you had, I was simulating, constant this stepping stone model, nice grid of populations. The direction was determined completely by the sampling variation. So if you want to interpret the direction, you need to show that it's showing somehow come up with a method for saying that it's stronger than sampling variation. So you can imagine maybe some bootstrap-based approach where you resample your data and see that it consistently comes out one direction versus varying uniformly in every direction like you might expect under this an explanation of sampling variation. So it's more complicated than people have thought, and I have been doing more work to look into what is the pattern of the PC1 gradient when there's a range expansion. Does it always consistently go in one direction? And that's actually ends up being an interesting study. So in many parameter settings, when you have a range expansion, the PC1 gradient actually ends up being perpendicular to the direction of the expansion, which is opposite of what the kind of intuitive approach was, even my own. I thought, oh, maybe in expansions it does line up. But it actually goes perpendicular, and that's interesting. We have some ideas of why that is.
I got a bunch of questions, but the first one, so your simulations assume that the isolation by distance things come to equilibrium, and you could probably calculate how long it would take based on some model of diffusion distance and such things. So I guess the question is how reasonable an assumption is that Europeans are in equilibrium from 10,000 years ago, say, or from 50,000,
Right.
18,000 years ago?
So I guess in some ways I'd actually take the question differently, which is that the simulations were done to kind of show that it arises in these isolation
distances.
Yeah,
but I guess put my question a little differently.
Yeah.
If there was a real expansion and then it started to be erased by the isolation by distance.
Yeah. How long would it take?
How long would it take?
Yeah. I don't have a good answer to that. Yeah. Okay, the answer I have is that we have simulated the Neolithic expansion.
Yeah.
And under many ranges of the parameter settings, you get these gradients arising, and in many settings for the parameters, you get the gradient arising perpendicular to what you see in the data. So, and then in certain ranges where there's admixture between the Neolithic expanding agriculturalists and the Paleolithic populations that preceded them, you can get it lining up with the direction of the expansion. But it's, yeah, so that kind of gets at it in some sense, that you see those gradient patterns even with the population, the parameters of a Neolithic expansion, which is a relatively recent one. Yeah.
In these simulations I've seen with this guy's name, I forget.
Scofier?
Yeah, Scofier,
Yeah.
and so on.
Yeah.
Did you show this sort of serial founding effect or serial founding effect with admixture?
Yeah.
They seem to tell a different story, or am I misreading?
No, so actually, yeah, the work that I'm doing, I have a couple slides if you want to see it. But
well, maybe you could tell me that.
I'll just tell you. So the work is with Laurent Escoffier's group, because they've paid the most attention to simulating the Neolithic expansion, and so we're using their parameter settings, working with them, and remember when I said that the PC1 gradient aligns perpendicular. Actually, our best explanation for it is the surfing phenomenon that they've been describing. So this phenomenon is that when you have an expansion, you have a well, there are different models of an expansion, but one that's believed to be fairly realistic is that you have a serial founder effect occurring, where as the new unoccupied habitat is colonized, you take a sample of a subset of individuals from the current population and move it forward, right? That leads to a kind of strong level of genetic drift because you're subsampling the population. And what can happen is that a mutation that's very rare in the founder population, because of all this enhanced genetic drift of multiple founder effects happening, can go to really high frequency in the colonized populations. And I can't resist, I'm going to show this slide, because this is a really cool experiment verifying this, because it's been described in a number of theoretical studies, actually first in a paper with Cavalli Sforza and his colleagues. And okay, this is an experiment that shows this going on, which is, it's a bacterial, there was a bacterial colony initialized here with two different fluorescent colored alleles. You can imagine one locus, two alleles, a red allele, a green allele. And so you've got a kind of mixture in the source population, and then the mat grows outwards. And as it goes, there are these serial founder effects as the bacterial mat moves outwards. And in some populations, the green allele wins. And once it wins, there's no polymorphism, so the genetic drift doesn't even matter. The green is just going to be fixed in all the descendant populations from that one. In other populations, the red fixes, and then red goes on. Okay? And so you get these patches or sectors and of homogeneity and allele frequency. And what's interesting is that this might explain why we see PC1 going perpendicular to the expansion, because if you look at this, what's the direction of most genetic variation differentiation? It's perpendicular to this expansion in that direction. So it's kind of a cool unexpected behavior, and relates to that story of how to interpret these PC1 gradients. So it's pretty cool. What else was I going to say there, though? Oh, yeah. A bigger issue, too, about this surfing phenomenon, though, that's worth knowing about, is that these, when alleles surf, they go to very high frequency very quickly, and they are highly differentiated between geographic regions. So these look like, these would show a lot of the signatures of selection that you would, or the population genetic signatures that normally would be ascribed to loci that have been under natural selection. And you might look at this and say, oh, there must be environmental pressure for red here and green here because there's no variation in either of the two patches, and you'd be completely misled. So that's yet another complexity to the modern endeavor to study natural selection and using population genetic patterns, is that you might have, and some people have claimed that some of the signatures of natural selection that are being observed are actually just alleles that have been surfing. So, yeah.
So suppose now you want to find out which aspects of human racial variation are like the green and red patches up there, and which ones are the result of some kind of selective process. Can you interrogate data sets like you have in order to determine that? Like, for example, are there genetic correlates of racial phenotypes that one could examine to see if there's evidence of selective sweeps?
So I'd say for distinguishing these different patterns, I think we still need more work to, at the broad scale, qualitatively, what we know is that they can look the same, and there haven't been good tests for telling apart an allele that's surfed versus an allele that's been selected. One thing that would help would be to have, for instance, looking at the geographic distribution, so that if you see that the allele that you are inferring to be under selection is distributed in a way that it is at highest frequency the furthest away from the direction of expansion routes, then that might be one where you have less confidence versus one where it's going, it somehow doesn't line up with the expansion route. So for instance, and because this is relevant to the BEC theme, the microcephalin and ASPM gene papers that this is Bruce Long. Bruce Lawn at Chicago found evidence for in two different genes that are involved in human disease phenotypes that are involved with like microcephalin. From the gene name, you can imagine it has something to do with brain morphology or head skull morphology. So it's a gene that is involved in a disease that is related to some kind of abnormality in brain skull development. And it shows a signature of selection where there is a haplotype that is at high frequency in East Asia, and this was put forth as representing selection related to learning or intelligence, because you have a signature of selection in a gene that's related to some kind of disease trait regarding brain development. So, but others have claimed that this is a signature of selection, or of surfing, that an allele that could have surfed, gone as humans expanded out into East Asia, it would have risen to high frequency just like these did, and that you can't claim that that's a signature of selection. So, anyway, so more careful studying of what the two signatures are of surfing versus a selective sweep would help. There might be a zone where you just can't tell the two apart, like in things that have this kind of distribution where it's exactly going opposite of the direction of an expansion. And then, you know, like a lot of the inferences we have to do in evolutionary biology and in historical anthropological sciences, there can be multiple avenues of evidence. So if it's a signature of selection that's where it's a gene that also shows a signature in multiple other parts of the world, where you see the sort of like lactase. So people could claim that lactase in Europe is an example of surfing because it's in highest frequency in northwestern Europe. It's kind of exactly in the direction of the expansion as well. But seeing the same gene having a relationship to the same gene having a relationship to lactase persistence in another part of the world helps build the story and make it a more credible example. Yeah.
I'm not sure I understand this phenomenon well that generates the disequilibrium signature for selective sweeps, but isn't it the case that you expect to see, because there's a sampling error, that the mutation originally arose in a single individual, and therefore the other alleles that are on that chromosome get carried along? That's the reason why you get this signature, right? Isn't that going to be centered around the locus of the mutation that's being selected for?
Mm-hmm.
Because the probability of crossing over decreases with the distance from that locus, right?
Right.
Is that also going to be true for these kinds of things? I mean, is it going? Are you going to see? I mean, when you see the allele surfing, is it going to be centered around particular loci in that way, or is it? I would not think.
Yeah. So the difference might be in the decay, but you would expect it to. It can be, if it's rare enough, it would be centered around certain locations. So the so think about it this way, that each section of the genome, and once you get far enough apart, they're kind of effectively independent from one another. And so this surfing process, if it happens very rarely, it will happen to just a few loci in the whole genome. And so it will be a localized signature. It won't be sort of something that's seen everywhere. That's really what has to be true for the two to be confused. And I think there is some debate that some people think, you know, if the parameter settings of human expansions are enough to lead to lots of drift, then you would see lots of drift across the whole genome, or not at all, that surfing shouldn't matter at all, because it's not that much drift. But it might be that to have this really be a concern, you're kind of in a sweet spot where it has to have a rare enough that a few loci are showing it, but not all. Yeah.
So trying to put this together with what you said earlier about a single mutation versus polygenic selection, it seems to me that reconstructing the history of the adaptation becomes problematic now. So take your case of skin, light skin pigmentation at higher latitudes, right? You said, okay, different alleles in Northern Europe than we see in East Asia. There are two different stories about how that pattern could arise. One would be populations moved into higher latitudes and there were independent selection events on those populations separately of one another. The other is that populations began to move into northern latitudes, and there was some variation that was present there that was being selected on. But then you got surfing, so that the individuals who populated East Asia just ended up with one of the possible ways of having light skin. So the variation wasn't novel in East Asia or in Europe. It was always there in the founder population. It just got surfed out in one direction and a different direction in the other. Is there any way to tease those two stories apart?
Right, so I think your point's true even if you don't have to invoke surfing, just that like, yeah, you're right, that where I was saying there were two novel mutations in these two areas, it's very difficult to distinguish that from that there was some low-level standing variation, and just by chance one gets picked up in one area and one gets picked up in the other.
Well, it seems that there's a little bit of difference, so that if there are different pathways to light skin, and as the population moves across a cline so that the selection pressures increase, you wouldn't expect, if there are no trade-offs and costs and benefits for the different pathways, you'd expect them all to rise in frequency, right, without the surfing.
The stochastics might lead to, if one's at a higher frequency, it might take off and win. So I wouldn't necessarily expect that both would go. But yeah, I think that I don't have sort of detailed thoughts on that issue. It's an area where this gets even more complicated. The standing variation, I guess in some ways the bias and where I think most of us are interpreting this as two mutations is that you have strong linkage disequilibrium signatures for those two alleles, and standing variation very quickly breaks, you don't have as good of a signature of the linkage disequilibrium pattern, because once a mutation, if it's standing, it recombines off onto many different backgrounds and doesn't end up having such a strong signature. So, that's really how you tell the two apart. And in this case, the thought is that it points more towards two mutations. But being definitive about that, like was it very low level? If it's very low frequency standing variation, it still hasn't recombined onto many haplotypes, and it's hardly indistinguishable from being two different mutations.
I had exactly the same thought that Dan had. I wondered if you looked at contemporary populations in Africa, looked for lactase persistence alleles in populations that had no history of dairying. And maybe if you could eliminate admixture by doing some other kind of PCA thing you do. Wouldn't that be one way to test it? If the standing variation story is right, there should be a low frequency
of it in
all
other
three
populations. Yeah, that's right. The geographic distribution of the trait outside of areas where it's selected for would be a good thing to look at.
I'm wondering if you can tell whether this assumption that all of those SNPs are neutral is true or not, is possible. If you just looked at the individuals from some particular geographic region where there might be a selective pressure, and you analyze each of the several 100,000 SNPs for variation around the SNP, wouldn't you be able to at least decide which ones might be under recent selection or associated with some gene close by that is under selection?
This is to distinguish between the surfing pattern versus the selection.
No, just entirely different. The assumption in your map,
Yeah.
in the very first map, is that those several 100,000 SNPs are neutral.
Well, there's actually no modeling in that. It's just this principal components analysis, so there actually is no assumption.
Well,
But
once you reach conclusions that the
Yeah.
thing is just the result of reproductive isolation,
Yeah,
relative reproductive isolation,
Right.
with distance being the key factor,
Okay.
you're making an assumption that those are neutral SNPs.
Okay.
So I'm saying,
Fair
can you
enough. Yeah.
test that
Yeah.
with kind of massive calculations that would be necessary?
Yeah, so the closest I did to it was more of a data test, which is, so you could, but I did do a large selection scan, identify all the loci that you think are selected, exclude those. And I did sort of a first pass version of that, which is to look at all the SNPs of the most differentiated SNPs across Europe and cut those out of the analysis. So for instance, lactase is not, the lactase region, which is highly differentiated across Europe because of the strong selective sweep in the northern populations, is not included in the data set that makes up that map. I've excluded those regions. That's even excluded the top, the very top, I think it was 1% or 0.1% for those maps. But then more generally, I did an even deeper cut, and you still see these patterns even if you look at some of the least differentiated SNPs. And it's really that you have these very subtle allele frequency differences across many, many, many different loci that accumulate together to give you the signature. So, but you do see it, you know, if on the converse, if you look at just the selected loci, you'll also get the map, right? Because there's enough signature in the way that they have, or not selected, which they have a highly differentiated loci. Those alone contain enough information to see the genetic relationship in general. Yeah.
So I want to back up to the more general point about the relationship between genes and language. And if I understood you correctly, so we have cases like Hungarian, where it doesn't look like the language barrier is making any difference at all. We have Switzerland, where at first pass it looks like language matters, but then when you look at it more closely, it's really geography, because the Italian Swiss speakers are in the south, right, and the French Swiss speakers are in the west, and that is what you're calling
more
language.
difficult to be, yeah.
Yeah.
When language and geography are perfectly correlated,
Right,
it's really difficult
right.
to
So,
to what's going on.
I mean, with the exception of cases like the Basque, where arguably there's a completely different history there than perhaps might be true in the case of the Hungarians. It really looks like the ethnic markers of language are not doing very much. It's really just geography that's doing most of the work.
I'm generally in agreement with that from all the patterns I've seen so far.
So,
But it'll be
there's-
interesting to see how that plays out in different parts of the world.
I mean, people have made a lot of hay about the relationship between genes and language, right? So if language really isn't doing very much, it's just geography, then we need to revisit some of those accounts.
It's gonna be interesting. I think I'd love to see this done on a more global scale and see how these patterns play out, because I imagine it's not necessarily the same. It depends on how language is structured with mating patterns, but at least in these, and this is looking at fairly fine scale differentiation too, right? In Europe, it doesn't seem to, like you said, much play apart from the Basque, and then the Sardinia, that's more about geographic isolation.
Right, sure. Water.
Yeah.
What if you took the individuals you excluded because they have a mixed grandparentage
Yeah.
and put them back in with some sort of indication of where they came from, and showed, if you're right, they should end up intermediate between the locations of the grandparents?
Right. And most of them do, so.
Oh, you did.
Yeah. So, an interesting thing is how to accurately infer where their two ancestors come from, because you have this problem, and this is relevant to what I talk about, the personal genetic ancestry testing, that if you use these kind of PCA-based methods, which some of the companies do, so like 23andMe, you can place yourself on a PCA map. And they note this, they're aware of this and make it clear to the consumer, but the position of an individual who is half Swedish and half Italian, they'll look Austrian because it's coffee between. And so there need to be more sensitive methods that tease apart the signature. That it's actually right, and it should be kind of obvious if you look carefully that if you had the phased chromosomes, one whole chromosome should show more markers that are higher frequency in Northern Europe, and the other chromosomes should have alleles that are more common in Southern Europe. So it's not one of these situations where the information's not in the data, it's about how we're looking at the data. And PCA lumps the two chromosomes together, doesn't think about phase, doesn't treat each marker independently, so it doesn't capture, it doesn't recognize those more subtle signatures.
All right, we're out of time, but thanks very much.